coming, everyone, especially those of you who came because I sent that notification out to CIEHS members. So I appreciate it. Of course, it's a pleasure to welcome Dr. Banrida Wahlang, who is an assistant professor in the Department of Pharmacology and Toxicology, to speak with us once again this year and hear updates on her research program. Dr. Wahlang came to the U of L in 2010 and did her Ph.D. in Pharm Tox with Matt Cave. And during that time, she contributed to 7 papers or as first author. So she really launched her career with great success. And then she completed 2 years of postdoc at the University of Kentucky. And she was there an author on 5 papers and 2 as first author. And then she did 4 years as a postdoc and NRSA fellow with the U of L Alcohol Research and Superfund Centers. And there she published 18 papers, so you can see the logarithmic progression of her publication record, with 6 as first author. She then served as a year as instructor in the Department of Medicine. And during that time, we collaborated on a project with Matt Cave in terms of high-fat diet and PCB exposures and low-fat diet and PCB exposures in mice and mouse liver, and a model of MASLD and MASH. She assumed her current tenure track position in July 2022, and I certainly believe that she's well on her way to success with the School of Medicine PAT committee. She's currently an author on a total of 56 publications with 12 papers in the past 3 years, and I did go into PubMed and update the CV that I received. She's presented invited talks at a variety of national meetings. She's clearly very organized and hardworking, and she's an outstanding collaborator. Banrida is currently PI on a DoD TERP grant, acute and subchronic exposures to dioxin and dioxin-like compounds in liver disease, the role of sex and age. She completed her KO1 award and has received CIEHS and U of L Research Core vouchers that continue to support her research, and you'll hear more about that today. In addition to her research, of course, Banrida teaches in a variety of courses, and she is currently involved with CIEHS as the leader of the Career Development Program for junior faculty. And she has served on a number of NIH and DoD TERP grant review committees, and is an editorial board member of the Journal of Nutritional Biochemistry. And of course, she does ad hoc reviews, including for molecular cellular endocrinology, which is near and dear to my heart. And I always appreciate her insightful reviews. She has mentored undergraduate and medical students and is currently mentoring 1 PhD student in her lab. So, Banrida, it is a delight to have you with us, and we're looking forward to hearing what you've been up to in the past week.
Okay. All right, well, thank you so much for that introduction. So I did give a grand round talk last year about the exact same time. And so there's only so much you can do in a year. So I apologize if some of the material that I'm presenting today might be redundant. But then also, like, so much has happened in a year, and I felt like how I felt last year and how I feel this year, and the times are very, very different, but I'm happy to be here still. So there's that. So today we'll talk about, again, talking about, how we've changed a lot in a year. So if you will watch how I talk about my work today, it might be a little spin on it, but it's essentially, I'm still an environmental toxicologist, but we'll talk about food contaminants and the current metabolic crisis. And so before I start, I just want to say that there's no relevant disclosures today. I do want to acknowledge funding agencies as well as different centers at the university for their collaborative support. And so learning objectives, since this is the Grand Rounds talk, we want to describe what food contaminants are and their behaviors as endocrine and metabolic disrupting chemicals, identify the liver-related consequences of exposures to these chemicals, as well as how disease mediators like biological sex, as well as dietary patterns, modulate responses to these EDCs and MDCs. And so Let's start off first by talking about food contaminants. And so, as their name suggests, these are things that contaminate a food, and they're essentially undesirable materials in our food that could get incorporated in the food that we eat, right from the agricultural and farming processes up to packaging, storage of the food on a shelf, at stores, up to when it's presented in a plate in front of us. And so there are different types of food contaminants, physical contaminants, which are tangible materials like metal, dirt, or hair, or what you have you. And this is usually because of carelessness in the food processing or food preparation process. There are also biological or microbial contaminants like bacteria and mold, and these are usually the main culprits of food poisoning in our day-to-day lives. There are also chemical contaminants. And so in this broad category of chemical contaminants, there are these chemicals which are generally recognized as safe, like food preservatives, food additives, flavoring agents. There's packaging materials too that also poses chemical contaminants. And there's another important category of chemical contaminants that is related to our research, which are toxicants. And so these toxicants are unique in a way that they're not intentionally added to our food, but they're present there inadvertently. And so by, happenstance or just by virtue of being there in the environment, if you will. And these include industrial chemicals like PFAS, polychlorinated biphenyls or PCBs, and dioxins, pesticides and herbicides. And so these are usually, used in agricultural processes, and by virtue of that, they do get incorporated in our food, unfortunately, as well as heavy metals. And nowadays, there's a lot more of talk going on about microplastics as well. So these are essentially forever chemicals, and they're also now known as everywhere chemicals because they're persistent in our environment, our ecosystem. They're present in our soil, our water, our food chain, and so they are also present in our food. And so how do food contaminants act as endocrine disrupting chemicals? So endocrine disrupting chemicals have been defined as any foreign chemical, exogenous chemical, or mixture of chemicals that could interfere with any aspect of hormone action. And these food contaminants, or these chemicals that I'm going to talk about today, are also able to interfere with, let's say, sex hormones receptors or sex hormones or the thyroid hormone. And so they do act as endocrine disrupting chemicals. But what I want to talk about more is about this hypothesis that was brought upon, I would say, about 10 years ago. It's called the EDC-MDC hypothesis. And this was needed because a lot of work that was looking at caloric balance studies were unable to explain these increased rise in obesity and the metabolic syndrome in the last 2 decades. And there was a really neat study that was done, I believe, in 2016, where this group from Canada was looking at these NHANES cohorts between 1988 and 2006. And for any input of calories or macronutrients or, time for leisure or physical activity, there was this unexplained rise in BMI between those two different cohorts, between these 2 different years in terms of BMI. And it was like about 2.3 units or so. So key opinion leaders in the field came together, or, discuss about these, what could these other factors be that could explain this rise in these metabolic chronic diseases, and environmental chemicals was postulated to be one of them. And so the term metabolism disrupting chemicals was then introduced, and it was defined as environmental chemicals that can promote metabolic changes leading to diabetes, obesity, or a fatty liver. And so in part, they could be the culprit or partial culprits for these global epidemics of obesity and the metabolic syndrome that we're seeing today. And so this idea that these chemicals, these industrial chemicals and pesticides are food contaminants has also been validated in numerous studies. So a number of researchers across the United States have actually gone to grocery stores and purchased products from the groceries, take those products back to the lab and measure these chemicals. And you can see here in this graph that there's a range of chemicals that was measured, and you can see here that DDT is one of the most prominent pesticides that was measured in these food products, and they include dairy products, vegetable products, fish, meats. It doesn't matter if you're, vegetarian or non-vegetarian, you'll still have these food contaminants in your food. And there are also significant I guess, presence of these industrial chemicals like PCBs and PFASs. And another study that was looking, I think this was from 2017, where they did this WHO global survey on human breast milk. They also found that levels of dioxins, benzodiazepines, as well as PCBs in human breast milk is still present at levels that are still not considered toxicologically safe. So there's this idea that these chemicals have been banned for over 30, 40 years now. So their levels are going down in the environment. And yes, they are, but they're still at levels that could elicit a harmful effect in living organisms. So there's that concern as well. And so again, the ability of these chemicals to exert these endocrine and metabolic disruptions have also been, shown and demonstrated in rodent animal studies. Epi studies have done a lot of work looking especially at, phthalates and polycyclic aromatic hydrocarbons, as well as pesticides, with these facets of the metabolic syndrome, particularly obesity and glucose metabolism. And you can see that sometimes these chemicals do everything, and sometimes they affect lipid metabolism more than they would affect glucose metabolism and so on. But these associations have been characterized and are well reported in literature as well. And so when we talk about the metabolic syndrome, we always, think about the constellation of factors that define it, like hypertension, high blood pressure, dyslipidemia, and obesity. But what I'm going to stress on today is the hepatic manifestations of these obesity as well as diabetes and these components of the metabolic syndrome on the liver. So I'm going to talk about steatotic liver disease, and I'm going to focus a little bit more on how these chemicals act on the liver as well as glucose metabolism. And so I just want to state here that when we're talking about these diseases as well, I'm going to show you a little bit of global prevalence to just kind of emphasize the significance of these diseases, but also that there are sex-specific prevalence. For instance, with type 2 diabetes, the general consensus is that males are more susceptible and usually at lower BMI thresholds. So what about steatotic liver disease? So this is broadly classified into two types. One is metabolic dysfunction-associated steatotic liver disease. It has a global prevalence about 25%, and even in the United States, that's about the same prevalence as well. So it is the leading cause of chronic liver disease. It's prevalent in males and postmenopausal females, and it's usually attributed or blame on, high caloric diet, sedentary lifestyles, as well as genetic predisposition. There's another subset of steatotic liver disease known as alcohol-associated liver disease, and this has a lower global prevalence just because, there's just only a certain population that consume excessive amounts of alcohol, and it's brought upon by this excessive alcohol consumption. But there's a new term now that has been defined within the last, I believe, 3 years, known as MET-ALD, which is a combination of metabolic dysfunction as well as moderate alcohol consumption that could lead to these hepatic manifestations of the disease. Now, another thing in terms of steatotic liver disease that we're interested at as a group is the steatotic liver disease that's brought about by these toxicants or industrial chemicals or pesticides or generally food contaminants, and this is known as toxicant-associated steatotic liver disease. And it was initially defined in occupational workers and industrial workers for obvious reasons, but nowadays, if you look at toxicant-associated steatotic liver disease, or like we lovingly call it, TASLD, it's also seen a lot of reports, a lot of TASLD parameters have been demonstrated to be associated with low-level exposures to chemicals, so even exposures in the general population. And despite these different causes of this steatotic liver disease, I do want to say that in terms of disease progression and pathogenesis, they're more or less the same, starting off with a normal liver that then gets steatotic with lipid accumulation, could progress to steatohepatitis, which is lipid accumulation along with inflammation, and if untreated, will eventually progress to fibrosis, cirrhosis, and hepatocellular carcinoma. So why does it matters? And I presented this last year, and I think I talk about, so this is the prevalence, the global prevalence of diabetes reported in 2021. And as you can see here, these darker red areas are the areas where there's a higher prevalence as well as incidence report. And what I talked about last year when I presented this map was I talk about how this general idea of Western diet and ideas about metabolic diseases being more of, Western country kind of diseases has really changed, because if you look at these global shifts here, you can see that a lot more of these disease prevalence are happening in even Southeast Asia, the Middle East, and even in the Northern African region. And if you look at mast cell prevalence and incidence rate, it's pretty much the same pattern as well. And so when you're talking about what are the causes for these global shifts, there's going to be a lot of complex interplay between socioeconomic factors and access to healthcare, as well as environmental factors. But there's also this thought process going on that perhaps this modern rise in processed foods, for instance, and along with poverty and food insecurity issues that some of these nations face, there's more reliance on these caloric dense but poor nutrient foods that eventually could lead to these incidence and prevalent rates. And I also mentioned that males and postmenopausal females may be more at risk for mast cells, but this a report that was done in 2021 looking at this global burden of disease, it actually showed that in terms of mortality, women tend to, their mortality rates tend to be higher in women with mast cell than men. And I'll talk more about what some of those reasons are. So it's not just about disease prevalence, but also about disease progression. So before I talk more about industrial chemicals, I want to talk a little bit on these GRAS chemicals, or generally recognized as safe chemicals. And so these are chemicals which are designated to be safe, right, and are designated by the FDA because they've been, used historically in food. And there's little scientific evidence that show that they have any toxicity, and so they do not essentially require pre-market approval. But there's been a lot of controversy, especially this year, in terms of GRAS chemicals. And I'm very neutral about, all this, but I always look at things from a toxicology perspective, from a toxicologist lens, if you will. So in terms of GRAS chemicals, some of these factors that have led to a lot of debate and controversy could be perhaps incomplete safety data, right? So because they're historically used, doesn't mean that, they could be safe. And a lot of times, a lot of these GRAS chemicals were designated by the FDA back in the 1980s, when we didn't have these problems of obesity and metabolic syndrome that we currently face in our population right now. And there's also this idea that companies, right, food companies are doing the self-policing for these GRAS chemicals. So there could be a lack of transparency in data and so on. And just to add more fire to that, there's been a lot of studies within the past 2 years looking at some of these GRAS chemicals, particularly sodium benzoate, which is used in a lot of everything, literally everything. I look at my daughter's foods, there's sodium benzoate everywhere, and I don't know if I should be concerned or not as a mother. But there was a study that was recently published looking at sodium benzoate, a GRAS chemical, a widely used preservative, and they demonstrated that sodium benzoate at these levels that are thought to be safe, for a daily intake, actually cause inflammation and apoptosis of beta islet cells, leading to a compromised insulin secretion. And in part, sodium benzoate, what it does is it acts as, it gets converted in the body to benzoyl coenzyme A, which then leads to benzylation of protein, which is a post-translational modification, similar to acetylation of proteins, and eventually lead to these drastic impacts. And so closer to home, one of my former colleagues, Loretta Jofflin, has also looked at sodium benzoate, and she had performed these studies in male and female mice that were fed either a low or high-fat diet, and she used the concentration of sodium benzoate in drinking water for these mice at levels that are recommended by the FDA, which is 0.1% of food weight. And she demonstrated that despite a lower high-fat diet, these mice tend to have some kind of metabolic disruption. For instance, in mice that were fed a low-fat diet, she observed that male mice had liver fibrosis, and there was this protein benzylation that we talk about. And in mice fed a high-fat diet, there was liver steatosis in male mice. And so it was exacerbating or almost acting as an agent that could exacerbate liver steatosis when given with high-fat diet. And so one of the models that have come to play when it comes to sodium benzoate, again, was this benzylation of proteins, but how this benzoyl coenzyme A could sequester all the coenzyme A that's needed for metabolic homeostasis, like TCA and beta oxidation, and that eventually could lead to compromised cell function. And I'm just going to show that little bit of data there, but we can talk more if you have questions about that. And I just want to switch a little bit and talk about other ideas that came up during that EDC MDC hypothesis that I mentioned earlier. And one of the things that was defined during the, with these EDCs and MDCs as food contaminants was this idea of obesogens. So obesogens are chemicals that promote or contribute to obesity development or exacerbate diet-induced obesity. So just remember that all obesogens are endocrine disrupting chemicals, but I'll show you that not all endocrine disrupting chemicals generally act as obesogens. And I want to draw your attention to this study. This is one of the first studies that I did. Dr. Kling mentioned to me about being in grad school, and this, I think, was one of the first research papers I've published back in the day. And it was looking at this particular PCB conjugate known as PCB 153. And I don't want to admit we use doses that were way higher than we would see in humans right now, but we did demonstrate that this idea that these industrial chemicals act as obesogens because it was able to exacerbate this diet-induced obesity, and the mechanisms included increase in lipid biosynthesis as well as decreased fatty acid oxidation. And another concept when it comes to these obesogens too is about these windows of susceptibility. So there was a study that was done again by one of the obesogen experts, if you will, in the field, where he looked at these postnatal exposures to tributyltin and demonstrated that prenatal exposures could shape stem cell programming in the adipose tissue. And these effects on fat depot weights that were increased with exposure were seen all the way to the F3 generation. So in these, the F3 didn't get any, exposure. It was just a transgenerational inheritance of that phenotype. And so the importance, I guess, of these chemicals and how certain windows of exposures could impact outcomes over the years. So I also want to talk about sex differences because this is another component of my research program that we focus on. So we've talked about food contaminants, we've talked about metabolic diseases, but what about mediators of these diseases? So sex differences in liver health and even in obesity is increasingly important because of a number of realizations that come over the years. So one of the things is that we all know that males and females are different, but biological sex could mediate liver disease, for instance, by this diagram that you can see here, where these authors did a great job of putting this together in terms of risk factors. So they said that, and with literature reports supporting this, that males and postmenopausal females are more at risk for liver disease or even steatosis and fibrosis, whereas premenopausal females are usually at very low or even negative risk. And one of the reasons for this is the protective effects of estrogen. Estrogen is a great hormone in terms of maintaining energy metabolism in females, helps combat oxidative stress, reduces lipid accumulation in the liver if there is a hit, and so on. And so some of these endocrine disrupting chemicals could interfere with estrogen signaling processes to induce those harmful effects. And there are other things too. So I talk about a little bit about how females may be more at risk for mast cell deaths than males. So one of the reasons for this is there is these sex disparities, for instance, in liver transplant, right? So when a patient's liver is not working anymore and they need a transplant, somehow there are females that tend to be put on the list way down than the males. And there's a number of reasons for that, including diagnostics and criteria for transplant that are also not sex specific that could lead to this, but also in women, once they develop mast cells at that postmenopausal stage, their progression of the disease is way faster than males. So, I get this question all the time: if women are premenopausal, are not at risk for steatotic liver disease, studies study it, but women are actually, may have worse fate in terms of disease progression than men. And so it's important to look into that. And also in terms of prevalence, while diabetes and steatotic liver disease may be more prevalent in men, obesity in certain countries is actually more prevalent in women. And this is, driven by cultural trends and other factors in those countries. But it's also important to look at sex-specific pathways because what might we identify in one sex could actually be used in the other sex as a therapeutic intervention, if you will, or look for molecular targets. And also, when we talk about sex differences, it's not just about mechanisms and progression, but it's also about how we approach treatment and care. And this was this really fantastic article that I read in New York Times 2 weeks ago to talk about how obesity is killing American men, because apparently men compared to women may be less likely to seek, healthcare when it comes to weight gain. They just become unconscious of it or don't think about it too much as women. And so they're less likely to go for weight reducing drugs, if you will, and other factors. It's why biological sex is important to consider. So another thing also that we want to talk about is these doses. So I mentioned a little bit about how some of our studies look at high doses. As we go more and more into research, especially I think in environmental health, people want to know what is relevant and what's going to kill them and what's not, right? So they're like, okay, if these are doing these to mice, it'll do this to us. So maybe not, because, we might be using very high doses. So we've come to realize that using relevant doses is important in our studies, but also looking at risk assessment rather than hazard characterization. So we know that the dose defines the poison. So anything that is given perhaps at a higher dose and concentration could elicit negative effects. But in terms of risk assessment, what we're exposed to every day, what are those effects? And also, we're exposed to multiple chemicals at a time. So we want to look more into mixtures versus individual chemicals, while keeping in mind that when it comes to mechanisms, perhaps individual chemicals studies are still important to do. And so one of the studies that we did last year was to look at these low doses of these dioxin-like chemicals. And this was funded in part by the DoD grant. So we look at these mixture of chemicals, PCDDs, PCDFs, and PCBs. And so those are the chemicals that I mentioned were detected in human breast milk at, still high levels that are not considered toxicologically safe. So we look at these mixture of chemicals, we took biological sex into consideration, and we did in a 2-week exposure study, because we were like, let's see, maybe we don't see anything at these low doses. And we actually found that when it comes to hepatic endpoints, we saw that in our females. This is in the absence of a high-fat diet or anything. We saw that exposure to these dioxin-like chemicals lead to an increase in hepatic lipids. There was an increase in de novo lipogenesis, as well as a downregulation of ESR1, the gene encoding estrogen. And so we saw that there was this accelerated dysregulation of hepatic homeostasis only in females. And we were really excited about the study, because we were like, this is the first time we were looking at these low doses of these chemicals. So let's see what's going to happen next. So we decided to also throw in diet and also look at longer term exposures or subchronic exposures. And so we had this new experimental design where we had male and female mice eat on a high fat or controlled diet. So this high fat diet is a 42% fat. It's high carb, high fat diet, and it's synonymous with a Western diet. So we call it synonymously high fat or Western diet. Sorry for the inconsistency there. And so we had these mice again expose these chemicals. So what we did in terms of doses was we went and looked at the CDC human exposure database that had measurements of these chemicals in human serum collected for I think since the 1990s up till today. Up till a few years ago. And so we look at the levels that were present in human serum at the highest levels, I believe, and then we extrapolated those levels back to what those will be in mice. And then we did these exposure studies, exposing them every week to see, what are the outcomes from these exposures, since we're exposed to these chemicals every day. And so what we saw was very highly interesting. So if we look at obesity and steatosis development, there's apparently sex and diet differences. For those of you that are not familiar with, sex and diet differences, usually with body weight gain, males on high fat diet or males in general tend to have higher body weights than females. And then in terms of the impact of high-fat diet, especially on the strain of mice, which is a C57 Black 6, males are usually for 12 weeks of high-fat diet feeding show more steatosis and obesity type phenotype, whereas females are more resistant to that. And that in part could be explained by what we talk about, that estrogen protection. But I just want to say that we have also done some studies, or other groups have done studies that have looked at high-fat feeding for a longer period of time, and actually saw that at some point the males and the females kind of look more or less alike, and those sex differences eventually disappear. But for this study, I want to draw your attention to these low-fat diet groups, because in the high-fat diet groups, unfortunately, we didn't see any dioxin effects. So we were bummed that dioxin was not a diet-dependent obesogen. But we did see that in a low-fat diet, especially in our female mice, they had higher percent fat composition relative to body weight. They also had significantly higher ALT levels, hepatic triglycerides, and there was an increase in CD36 gene expression, which is an important fatty acid transporter in the liver. And so these data, along with our histology data, which also showed that our I don't think you can see it very well here, but like our female exposed mice tend to have higher frequency of lipid droplets in our female controls, showed that these dioxin effects were only seen in the female mice, but also on a low-fat diet. So we concluded that, all right, dioxins are not diet-dependent obesogens. I mean, we're not necessarily surprised by this just because of the way that dioxins act mechanistically, but we were also somewhat, wondering why are these females more susceptible to this. So we thought maybe males are protected. Unfortunately not, because when we look at glucose uptake and insulin resistance, we saw these dioxin effects largely in our male mice. So we did a glucose tolerance test at the mid-study period as well as the end of the study, and you can see these are females. So the area under the curve, so these curves over here between the exposed and the unexposed in both diet groups are kind of pretty tight. But if you look at our males, they, especially in the low-fat diet, dioxin exposure tend to increase the area under the curve. We measured insulin levels, and we saw that there was a drop. It was not significant, but it was a drop in insulin levels in our low-fat diet exposed male group. And so we did HOMA-IR and HOMA-beta calculations and actually found those significant effects. So this shows that while these dioxins appear to impact lipid homeostasis more in females, they tend to impact glucose metabolisms more in males. And we don't really know why, but we have some hypotheses about that. All right, so another thing when it comes to dioxin exposures and dioxin studies and talking about the relevance of these findings, we want to keep in mind that dioxins are, for those of you that are familiar, know that they are AHR activators. They activate this aryl hydrocarbon receptor. And the aryl hydrocarbon receptor is unfortunately a double-edged sword. So, physiologically, it's important for you. It's important for organ homeostasis and cell cycle, and plays an important role in nervous system, in the liver, and the immune system. And in fact, a lot of studies have looked at AHR microbial metabolites and demonstrated that these metabolites play an important beneficial role in the gut because they modulate immune response. But when it comes to toxicity, the studies, the 4,000 papers that have been published so far in terms of dioxins show that the AHR is bad for you. So there is this, conundrum. So is AHR good or bad? And so I work with Dr. Krishna Jala on this. He works with AHR activation by microbial metabolites, so these low affinity ligands, and demonstrate that they're good for you. So they're beneficial for you. But when you're thinking about dioxins that are, really good AHR ligand binding compounds and that are present in our body continuously, and there's this persistent activation of the aryl hydrocarbon receptors, perhaps could lead to toxic effects. So when it comes to AHR, there's another thing that's a problem and a controversy in our field. There is this human versus mouse AHR debate. So the ligand binding pocket of the human AHR has been evolved over time to be more, complementary to endogenous ligands like these gut microbial metabolites, whereas the murine AHR is not necessarily so. So a lot of these studies that have looked at dioxins, at these toxic exposure, toxic equivalency factors, have based a lot of their work looking at mouse AHR, at mouse cell lines. So it's still an ongoing debate. And we wanted to address this head on. So I do want to show you here that like the human AHR, and this is the mouse AHR allele in C57 black 6. So this ligand binding domain over here is different, whereas some of the domains are conserved. And this is another mouse AHR allele. It's expressed in DBA mice. It's also much more similar to humans. So if you give dioxins to a C57 black 6, you might see, chloracne or some other drastic toxic effect, but you may not necessarily see that in the low affinity allele in the DBA mouse. So we wanted to address this problem head on. Fortunately, with the DoD grant, we were able to get some money to get these humanized AHR mice. So we got these from Taconic, and I just want to say that there was a group that attempted to generate this humanized AHR in 2010, I believe, and nothing. I don't know what happened. There's nothing in literature, so I don't know what happened to that group. I should reach out to them. But Taconic generated this humanized AHR model where they took they did a targeted replacement of the mouse AHR exon 3 to 11 was deleted and inserted with a human AHR gene. This represented this humanized AHR that we'll be using in our study. And so they did keep exon 1 and exon 2 of the mouse because this, in terms of species differences, is very minimal. It's very conserved between mouse and human. Exon 2 is also the domain, the exon that encodes for the DNA binding domain in the AHR gene. And so, so what did we do? So we took these humanized AHR mice. We did that same exposure. We started off with a 2-week study because, this is the first time we're doing it. We weren't sure if these mice, how are they going to behave, or are they going to be healthy at all? But they look great. So so we know that that humanizing these mice with the AHR is okay. So we did this 2-week exposure study, and we first looked at AHR target genes. So we first validated that it's the human AHR gene and not the mouse. So the data here is from the mouse model, and this is from the humanized model. But if you look at our AHR targets over here, you can see that there was really no induction in these AHR target genes like cytochrome P450 1A1 and 1A2. And so we're sitting here thinking perhaps our hypothesis is right. So maybe the human AHR is not really, responding to these dioxins at low levels. But I want to take these results with a grain of salt because this is a completely new model, and we still need to first demonstrate that it's actually a legit model. But I thought I would just share these data here anyway to kind of, show you that perhaps these low-level dioxin exposures may not be activating the human AHR and not elicit those steatotic and glucose metabolism toxicity that we saw with our mouse model. But alas, that's not the case. So we look at our glucose metabolism, and you can see here that compared to our human compared to a mouse, there's still these similarities in terms of glucose tolerance tests, especially with plasma insulin. So there was this trend in our plasma insulin levels in our mouse AHR model that was significantly reduced in our human AHR models. So this made us think that perhaps these effects may not be canonical AHR-driven pathways, but it could be a direct dioxin effect on pancreas. Yes. Do you have a question? Yes.
Liver specific?
It's a whole body. Yeah, no, a whole body. Yeah, yeah. So we don't have. I know. It's a whole body. So we did determine that it is a whole body global humanized model, and so we did see these effects, which made us think that perhaps again, these may not be AHR driven pathways, or they could be non-canonical AHR pathways. So we're still in the weeds of this and trying to figure that out. But I also want to show you another. So hold on. So our humanized AHR study suggests, like I said, that there could be these non-canonical AHR effects. But we also saw that pancreatic toxicity. And I said, it's so great. I'm doing this grand rounds. Anybody from endocrinology that wants to collaborate with me to look at the pancreas, please feel free. We really need some help because we have seen this effect with these dioxins and even with our PCBs that they're decreasing insulin levels. They're enhancing. It looks like their quickie results look great. So it looks like they're very sensitive to insulin, but then their glucose, tolerance test curves are just way off, which makes us think that perhaps there's a direct effect on the pancreas. So yes, so would like some collaboration over there. Okay, so what about other longer term exposures? So I've shown you a 12-week and a 2-week exposure, right? So we wanted to know, what if we expose these dioxins and then supposedly we stop being exposed to them? And so this was also based on the DoD grant, which was reflecting military exposures where troops would go out, be exposed to these chemicals and come back home, and their exposures would be way down from what they're exposed to in the military. And so we are still continuing these studies, but I also, again, want to show you the glucose tolerance test and how these effects could, just without even the exposure, how physiologically, age matters and time matters. Because if we look at our 12-week period over here, we did see sex differences in our glucose tolerance test and maybe, again, those dips in GTT with these, with 12 weeks. But then here we kind of see that the sex differences effects have disappeared. And so we are still doing this study. We are going to look at, I think the study will be completed in February. So we are excited to look at those livers and see what's happening there. But yeah, that's all I can share for now in terms of our long-term exposures. And another paradigm that we're thinking about too in terms of exposures is, what if we introduce a high-fat diet after the exposure period, and what would that do to, the liver or obesity or glucose metabolism? So when we get the money one day, we will hopefully do those studies as well. So, okay, so I just want to wrap up here and get some, give out some concluding remarks. So why do these studies matter, right? On food contaminants. So in terms of dioxins, there's no regulation by the FDA on how much dioxins is there in our food. There's no ADI. There's nothing. The European Food Safety Association, EFSA, is actually way better because they have some recommendations for maternity diet and so on in terms of dioxin levels, but we don't. But even in terms of grass chemicals, I talk about, how these designation were given back in the day, and a lot of things have changed. Right now, we have better studies that could mimic or reflect those longer term exposure, postnatal exposures, and so on. So I think it's important for agencies out there to take these into consideration. We have AI that could help put all these studies together. Why not use it for the good and give us better information on how bad or how good these chemicals are and what levels should we incorporate for our public safety. I also had another thought about what about grass chemical plus these unintentional contaminants, what those things do together. So, let's say sodium benzoate and dioxins, or another food grass chemical with PCBs. So again, we don't know what those effects could be, but that's just something, food for thought, that you want to think about. We also want to think about childhood exposures, and this should be transgenerational. I don't know why it says transgressional. Maybe it should be transgressional. Transgenerational inheritance of disease. I showed you that beautiful data with that obesogen tributyltin, but and there's a lot of transgenerational exposures that has been done with PFAS and PCBs and so on. But what about these low dose levels that we are talking about? So what about those actual levels that are present in our human blood, and what are those effects? So we don't know that yet. And then again, lifestyle factors. Diet was one of them, but what about alcohol and smoking and vaping, for instance? I see someone in the room that does that kind of research. And what about biological mediators? We talk about sex differences, but again, in terms of sex differences, one thing I want to talk about when I'm concluding is, we talk about this general paradigm that males and postmenopausal females are more at risk. Our studies, so this is just a subset of our studies, but our other studies on pesticides actually show that these chemicals are able to interact with sex hormones like estrogen and androgen, and actually it suppresses estrogen signaling. And these are in adult mice that are, representing young adult women and not postmenopausal. And so what if these endocrine disrupting chemicals are actually flipping the narrative that perhaps when you're exposed to these chemicals, you're compromising your endocrine system, your estrogen protection, and you may actually be more at risk. And perhaps that could be one of the reasons that maybe 40 years from now, we might see these prevalence changing. So, yeah. And then nutritional and intervention strategies as well is part of what we're interested in. So with AHR, we talk about one of the things I'm working on with Dr. Jala is urolithin A, which he's been using for everything, if you're familiar with his research, and it seems to protect everything. So what if it could also protect against dioxin exposures? And so with that, I just want to close, and I'm happy to open any questions, take any questions, and yeah, thanks for listening. And I do want to thank my team, my students, for all their work on the animal studies, and my collaborators and everybody. So yeah, thank you. Yes.
You best stage your minds, but I didn't catch how you use that information. Do you only euthanize at a certain point of the cycle, or you just use it as a covariate?
So I didn't show it with this study, and we have the data too. So we monitor estrous cycle is that what you're talking about, right? So we monitor estrous cycle 5 days a week once we start the exposures. And we've seen that with the with PCB 126 and chlordane, that the mice are exposed actually have a prolonged estrus phase, the more cornified cells that we're seeing histologically, than with the other unexposed mice. So that's one way that we monitor it. And we also did some studies using overrectomized mice, and we've seen some findings that have somewhat supported our mechanisms. So our exposures didn't really do all those, increase in liver weight and all that with our OVX model as they did with our normal mice. And that kind of told us that perhaps this mechanism of suppressed estrogen signaling is not in place. So
more to see if there's taking tissues at a particular point in time.
we could do that too. So, but we haven't done that. So yes, that's been asked before. It's hard. I think it's something, you need a lot more animals for that.
Yeah.
And yeah, and we're right now, I think we're just characterizing the effects, but yeah, but I think we were planning to do that with our chlordane studies, and just waiting for funding to get those done. Yeah.
On this, but I've been thinking, beginning, how are you going to, or how do you think you can parse out I'll say they're all chemicals, but let's say the chemicals that you're getting through foods that are coming from the ground or whatever, such as dioxins, PCBs, that's what we think about, as opposed to what are actively added food additives. Because things like the food additives would be at a higher concentration. And along with that, do they have similar data in terms of the contaminant levels in foods in Western European countries? Or is all that data from US foods?
It's both. So for instance, with sodium benzoate, I shouldn't say this, I think this is recorded. But anyway, so for instance, for example, with sodium benzoate, it used to be 0.1% or 5 milligrams per kilogram body weight. That was the limit that was set in the 1970s or 1980s. Sorry, 1996. But because there was a study done by I think WHO that looked at children that up to the 95th percentile, they were consuming up to 20 milligrams per kilograms. So just because they're consuming that amount, they changed this limit up to, and then the FDA sort of followed along with that. So I think the data is based on not just US, or, it's just whoever gets it first. And then based on that.
The European Union, they don't have as many additives to their food. I was just curious if maybe their contaminants are also lower.
Oh, you mean in terms of lab? No, I think So so it depends on pockets and regions. So I think, for instance, with PCBs, there's a lot of PCB pockets, like areas like Anniston, Alabama, for instance. I think in Europe there are also certain pockets, and they do base their data on that. But Europe also manufactured PCBs, right? Asia also manufactured PCBs, and these chemicals, they migrate. So it's not about, if it's less there, and that's why they have less restrictions. Uh-huh. We've not done that. I think that would be something to look at in terms of how much is present in the soil and these obesity and rates and so on. Yeah. Oh yeah, I know. I know. And again, and we shouldn't be the one that have to deal with this, right? Like, we're paying for food anyways. I think it should be, the higher ups that have to take the pain to say that this is safe for you, and then we take it. Like, why is it on, Healthy Moms USA to look at all the labels and then come up sometimes with even, wrong information or misinformation that, that, for instance, what happened with the Girl Scouts cookies, right? That they're bad for you because they have certain chemicals. But when you look at it, it's like, if you need to eat 60,000 of those Girl Scout cookies to get those chemicals in your body.
Well, a lot of these chemicals, that's why, so what you have and what you have.
Yeah.
So is this the broad category when you say PCBs and specifically, does it matter which one?
Yeah, so they're like, so depending on the chlorine substitution and the biphenyl ring, there's about 209 congeners, that you could make, but I think about 130 were made back in the day. So a lot of the lower chlorinated ones, they can be metabolized and degraded, but the heavily chlorinated ones, even in the body, there's a lot of steric hindrance that doesn't allow the CYP enzymes to attack them and break them down. So those are the ones that are usually, that we talk about in terms of our studies, because they bioaccumulate. Yeah, and there's certain differences in receptor activation as well.
So on the,
Mm-hmm.
so were they less insulin resistant or more sensitive or?
They look like they're most sensitive, but the problem is they don't make enough insulin. That's what it looks like. Yeah, from their HOMA-B, data. And we've seen this a lot more with our males than our females. Yeah. No, I think we have histology samples if someone could look and see, but yeah, that would be helpful. Yeah.
Brain question, getting back on the benzoic acid topic, if you look at a label, it's almost always there.
Yeah.
And it's in a package. And something that's crossed my mind before, if you have that in the presence of ascorbate and some metals like copper and iron, you can get decarboxylation, you get benzene.
Mm.
So, but I've never seen anything where is it a source of benzene exposure? And benzene's pretty nasty.
Yeah, but no, it's just different. I think it's because it's in diet. And as soon as it gets in, it does get turned to benzoyl coenzyme A. So no. Yeah. No. And then the body has ways to remove benzoyl coenzyme A, but sometimes with these chronic, everyday, dietary intake, that could, saturate the excretion pathways, and then eventually leading to those benzylation or whatever you call them. So, but I think with sodium benzoate, like, a lot of papers have also looked at sperm function. So there's a lot more on that as well, and steroidogenesis. Not a lot on obesity and liver disease. It's new, and in a way, it is controversial. I mean, because we have benzoic acid and sodium benzoate derivatives everywhere. So, but it's important, and you think about, kids eating everything, and it's everywhere, and then when they grow up, it's going to be of them. We don't know that. Any more questions? Oh, yeah, I don't know how to do that. Click continue. Yeah.
Well, I'll make a comment.
Question
Something
here.
that I read recently in a non-scientific journal, National Wildlife Magazine. But it was about, contamination with DDT in Arkansas. I couldn't believe the levels, and what it turned out to be is that farmers, because DDT is such a great pesticide or
Mhm.
insecticide, shall we say, they're still buying it through Mexico to spray on their fields.
Mhm.
So some of these chemicals that we think
Yeah.
can't possibly be coming into our food
Mhm.
system
Mhm.
chain may be.
Yeah, and I think
Those
no, I think I've heard about that, the DDT smugglers. But yeah, but I think DDT is just, I mean, it's just
Yeah.
out there, and I mean, a lot of countries use it till today, and in
Yeah.
China and different parts of Asia, and it's and it's effective. It does its job, those, yeah.
Despite Silent Spring in 1962.
I know, yeah.
There
Alright,
is
we
this
good?
question on from Dr. Winters about AHR knockout.
The level, we haven't looked, could I just type?
Now you can answer
Oh,
them. He'll
okay.
be there.
All right.
And,
Yeah, so we haven't looked at the done any Western blots to look at protein levels, but we did look at gene expression levels, and they seem more or less, the trends are more or less the same.